Welcome back from the break, everybody. Thanks for joining us again. We are moving straight on to our second session of the day, which is our short talk session. Again, please keep the questions coming and we will answer them all at the end with all the speakers. So this afternoon's talks are themed around promising new approaches to therapeutics and platform technologies. And we've got some great speakers lined up. We'll be starting with Felix Richter, who's been looking at the relationship between autophagy, inflammation, and lipids in aging. So with no further ado, I'll hand over to you, Felix, to get us started.
Thank you very much. Let's see whether I get this to work. So, yes, thank you very much for the kind introduction. My name is Felix. I am a PhD student in the lab of Katja Simon here at Oxford University, and I'm about to finish my PhD. So Friday is submission time for my actual PhD. And now I will present you today a little bit about how basically my PhD project has been sort of like translating into a more aging-related topic, which is basically the role of autophagy, a cellular process, in adipocytes and how it can impact sort of on lipid metabolism and how this lipid metabolism in its ways can control health. To start with, I wanted to kind of like give you a short introduction into aging. One of my colleagues later on, and I think on who's going to speak on Thursday, she's going to probably talk about this again, but just to get a small sort of like introduction into the process of autophagy. Autophagy is an evolutionary, very well conserved process. It's basically started, find it already in yeast, but it's really well conserved until human as well. And it's very essential for cells in order to kind of like get rid of rubbish and to restructure the cell in order to make maintain the cell in its own ways. And in its ways, I'm going to try that with a laser pointer. In its ways, autophagy basically is forming these double membrane structures, which will engulf a specific sort of like cellular cargo, and using a very dedicated, as said, evolutionarily conserved molecular machinery, it can basically engulf the cellular contents in its entirety, and then lead them to degradation through fusion with a other cellular component, which is the lysosome, which is an acidic organelle that basically helps to chop down these bigger macromolecules into smaller bits and pieces that then can be recycled by the cell in order to use them either for other biological processes or for energy generation, or even in order to provide sort of like these metabolites then to other cell types surrounding them. One of the things that I want to highlight here already a little bit, because we kind of like going to come back to this later in this talk, is sort of like the necessity of these molecules called LC3, which is a typical sort of like marker for autophagy that we're using. It's typically found in the cytosol just, but when we have activation of autophagy, this LC3 gets lipidated and sticks then into these double membrane structures. And basically, when we're inhibiting the fusion with the lysosome, we can get this very nice accumulation of autophagosomes, and with that, these LC3 molecules as well. So with that, we can measure sort of like autophagic flux very nicely. One of the things that comes with age is that this process seems to be slowing down in a lot of different cell types. We have been investigating that predominantly in immune cells, but we did find that also, for instance, in other cell types as well, or as described in other cell types as well. When we now look into the adipose tissue, however, well, basically during aging, the adipose tissue undergoes quite some transformation. It becomes somewhat more chronic. We have the definite shift in the immunological phenotype that we find inside this tissue. So immune cells, especially macrophages, are becoming more pro-inflammatory with age, and this leads to sort of like adipose tissue dysfunction. It can lead to the development of other comorbidities such as type 2 diabetes or cardiovascular diseases. And so this is sort of like underlying a lot of like the metabolic dysfunction that we are seeing in age as well. So one of the kind of like goals that we were thinking about is kind of like maybe if there is a change in autophagic flux at the adipocyte side, maybe we can sort of like impact on the immune cell infiltration and make sort of like the tissue more healthy, and with that sort of like reduce the risk for other types of age-related diseases. However, this link between autophagy and the aged adipose tissue is still not very well established, and so we were quite keen to study this role of autophagy in the aged adipocytes. So in order to do this, what we really thought to do in the end was address three major questions. The first one was really just very simply asking whether autophagic flux and autophagy in itself is actually altered in these adipocytes during the aging process, and we want to study that both in mouse and human. Unfortunately, for the human part, we haven't had yet the chance to get enough samples to kind of like present anything here today, but we have started looking into mouse tissues. And so, as I told you, one way for us to measure autophagic flux is looking at this accumulation of this marker molecule called LC32 when we stop lysosomal degradation. And so basically here you have the DMSO condition, which is basically you have still functional degradation of autophagosomes, but then we can also inhibit here in the combination with different inhibitors, we can inhibit the degradation of LC32, so we get this accumulation. And as you can appreciate, you get a slightly higher accumulation in the young mice. And when we actually quantify this autophagic flux, we do find that this one is reduced in old, similar to what we have basically seen also in immune cells in other cells before. There's two more questions, however, that we really wanted to address and where it becomes a bit more therapeutic. So we wanted to know which kind of like lipids are actually released in an autophagy-dependent manner. And thirdly, we wanted to know whether if there are specific lipids that are released in an autophagy-dependent manner, whether supplementation of these lipids could, for instance, help to overcome or shift sort of like the macrophage phenotype from a pro-inflammatory to a more anti-inflammatory phenotype, and with this kind of like dampen chronic low-grade adipose tissue inflammation. So one thing we first of all sought to do is kind of like to measure whether autophagy in itself is actually important for the output of free fatty acids or fatty acids into their environment. And so we stimulated from young animals, either wild type controls or where we have blocked autophagy specifically in the adipocytes, whether they can basically secrete less free fatty acids, and indeed this was the case. And this is actually fairly reminiscent as well to what we see actually when we compare this to young versus old animals as well, where you have about a 20% sort of like reduction in the total free fatty acid output that these adipose tissues can generate. So going forward from this, we wanted to next investigate whether there are specific free fatty acids that might be differentially downregulated based on the loss of autophagy. However, when we did this lipidomic studies, we did find that several of these free fatty acids seems to be reduced, and this seems to kind of like not have, the autophagy does not seem to have a very specific sort of like, or specificity for one specific free fatty acid in this context. However, we're currently are doing the same analysis basically for aged mice in order to compare and correlate this and see whether indeed autophagy could be a major role in and show us a similar profile. We next wanted to go on though to see more functionally whether free fatty acid availability might be altered, might be altering macrophages in aged adipose tissues. And one thing that is very nice, a very nice correlative for free fatty acid availability in mouse and humans actually is the expression of fatty acid uptake transporters. So what we did is we looked into adipose tissue macrophages and looked at the expression of a particular free fatty acid uptake transporter called CD36. And as you can appreciate here is like we got actually a reduction in the old mice in comparison to the young in the expression of this uptake transporter, sort of suggesting to us that free fatty acid availability also in vivo is actually reduced in the aged adipose tissue in itself. We next kind of like went on to look whether the expression of this fatty acid uptake transporter might be correlated to the production of different either pro or anti-inflammatory cytokines. So as an anti-inflammatory cytokine, we have IL-10. Otherwise, we have here in red a pro-inflammatory cytokine called TNF. And while CD36, again, very nicely correlates with free fatty acid availability, it also correlates with the production of IL-10. So with high expression of CD36, hence also higher free fatty acid availability, we did have higher expression of IL-10 as well, whereas this was not very much impacted in the TNF condition. So the last thing that I wanted to present to you today quickly is we were actually sort of like aiming for identifying whether there are specific free fatty acids that might be able to be used, or lipids that we can use in order to shift a macrophage phenotype and promote their production of free fatty acids, anti-inflammatory IL-10. So what we did is basically we used macrophages that we took from animals, and we supplemented them with free fatty acids of different kinds, and then we looked into the expression of IL-10. And so this is sort of like shown on this right-hand graph. So in gray, you have lipid-rich medium, which serves as a baseline control. And as quite nicely as you can see, is like when we look at the production of IL-10 from macrophages that have been cultured in lipid-deprived medium, we get this stark reduction in IL-10 production, which is kind of like very much in line with what I showed you just previously. However, when we, for instance, added back a specific free fatty acid such as linoleic acid, we could actually fully restore the production of IL-10 in this context again, sort of like suggesting that potentially specific free fatty acids could be used in order to promote a more anti-inflammatory phenotype in also an aging type context. And with this, I want to kind of like already come to a close, where I'm going to just highlight it to you today that the autophagic flux in aged adipose tissue seems to be reducing, similar to what we have seen in other tissues and cell types before. Similarly, we find that free fatty acid secretion, reminiscent of what we see in autophagy knockout adipocytes, is also reduced from in aged adipocytes as well. And lastly, we do find that local free fatty acid availability and potentially here linoleic acid, although we want to kind of like do some more experiments to confirm this, indeed seems to be controlling the secretion of IL-10 from macrophages. So what we really want to take this forward is in future, we want to kind of like continue and expanding the study predominantly now to human as well. And we want to kind of like correlate it to the free fatty acid secretome and test further sort of like lipids and free fatty acids that might be used sort of to dampen or at least help reducing inflammation in this context, and then try this in aged animals as well. Lastly, I want to just thank our group and especially UK Spine for the funding as well. And especially I want to also thank Irina Gucina at Cardiff University, who helped with a lot of the lipidomics analysis in this project. Thank you.
Thanks so much, Felix. That was a really nice story. So again, we'll move on quite quickly. We've now got Carl Morton joining us, who's at the University of Oxford. And Carl is going to speak a little bit about a project he's been working on through the UK Spine to explore mitochondrial function in different models of aging. So if you're ready, Carl, I'll pass over to you.
Okay, thank you. Let's try and share my screen. Just trying to see the share screen function. It's got a line through it, I think, on my... Ah, there we go. Entire screen, share. Okay. Can you see my slides okay? I can't see anything now, so I'll assume, just come back again. Yep. Leah, can you see those okay? Yep, great, sorry. I wanted to make sure after poor Thomas's first presentation. So thanks very much for the organisers for the invitation to speak today. So I'm based at the University of Oxford, though I haven't been there for quite some time with the pandemic, so it's quite interesting working from home most of the time. So this is a project supported by UK Spine with significant contributions from Hariba Scientific, just go to the raise pointer, who have been really helpful in keeping us sane during lockdown and also supporting us more recently when we started our work. So it's pretty easy to study mitochondrial function in cells, or either cell lines or cells from blood, but it's very much more difficult to do this in tissue. So if you want to study mitochondrial function in a tissue, it's much harder to get a handle on it, and you have to basically extract the mitochondria. And so it makes it very difficult to do that in certain tissues because they're not always easy to do. And also blood cells as well, you can look at, but you have to change the conditions. So we wanted to find a way of doing this, which was potentially much easier, and it could be applied to the situation in vitro, but also in vivo. If you had a sort of a tissue biopsy or some material like that, could really get a handle on what the mitochondria are doing. And this is also important for any therapies that are targeting mitochondria to get a handle on whether they're working. And also drugs. Many drugs are toxic to mitochondria, and in aging, drugs are a very important component of where people are going to try and target aging. So it's quite a significant process to do. So in terms of the project, so my lab work on mitochondria. So Sarah, who's been running this project for the last year, has been doing some great work recently with the techniques I'm going to talk about in a minute. So mitochondria can exist in many forms in the lab. So there's just some examples here. These are human fibroblasts, so they're quite filamentous, to see in green here. And these are some human hepatocytes where they're much more punctate. So they can look very different when you look at them in the lab. And also, my group are very interested in what mitochondria are doing in cells. Are they all doing the same thing? Or do you have specific mitochondria that do specific tasks at a particular time? And I'll maybe mention that a bit more briefly. And we're really, and we're really lucky to work with a fantastic group in Oxford led by Wei Huang. We've worked with Wei's group for about 3 years. And Wei is a bit of a world expert on Raman spectroscopy in applying it to biological systems, mainly working bacteria, but we've more recently started to look at eukaryotic systems. And Yabao is the PhD student, now the postdoc, who's worked with us on some other projects, and so she's a real expert on this. This is a setup. It's a confocal type microscope system with a Raman laser included. And this is just an example of a cell. So this is a PBMC hit with a laser, and this is what it looks like down the microscope, very different from what you'd normally see in a standard setup. And Hariba have been fantastic in this proposal. So Simon Fitzgerald is a sort of world leader in Raman and the physics of it, because obviously we work on mitochondria. It's quite different from what we know about. And then Nathan and Adam have also been involved in the project. Adam's more instrumentation, and Nathan is working in the labs at Hariba, and they're based in Northampton, which is very close to us. We've had quite a good interaction with these people over the time. So what is Raman spectroscopy? It's probably, I haven't got enough time to really go into any detail and really if you talk to the experts. There'll be a reference down the bottom from Yabao's recent paper on cancer. So basically, you excite your material with a laser, and then when you get what's called Raman scattering. So bonds, the carbon, hydrogen bonds, benzene rings, different molecules within that material will scatter the light, and you pick this up as vibrational energy. And so if you're looking at a biological system, what you tend to find is you get different peaks that contribute from different things. So there's proteins, lipids, nucleic acids, things like glycogen can have a strong signal. And for a biological sample, we tend to see two regions. So this larger region over on this side, where the bulk of things come up. And then over on this side, there's a specific group of bonded structures that come out. And in this middle region, we have what's called a silent region, where biological material shouldn't come up. And so we're looking at whether we can use these profiles to give us a handle on what a cell's doing, and maybe what a mitochondria is doing. So this is the paper that Yabao's just published, so well worth a read. I'll tell you a bit more about how you apply this technology to biological systems. And so just to give you an example, we've spent a lot of time over the last 3 years really trying to use this technology as a diagnostic tool. So we've worked in chronic fatigue syndrome, which is one of our main research areas. And we published a short paper in 2019, just going through a very small number of samples, just whether this was an approach that could be applied. And just to take you through this is new data from our group. So we've looked at 3 cohorts here, looking at the peripheral blood mononuclear cells. So the blue ones are from the ME patients, the fatigue patients. Multiple sclerosis we added in as another cohort, and the healthy controls. And on the left-hand side, you can see the Raman spectra. So these are average spectra across all the samples. The green is the controls, the red the ME, and the blue the MS patients. And they look quite similar. But we've applied a series of machine learning approaches to look at this and break these down. There's over 1,500 peaks here in a sample, and obviously by eye it's very difficult. And what was quite surprising is that we are able to resolve these groups. So we can separate out the healthy controls from the ME patients. And what was also very surprising was the multiple sclerosis patients also come out as different. So there's something clearly different between these 3 different conditions. And what's also interesting, that I haven't really got time, is the ME patients, going on Thomas's talk earlier, they actually separate out the severity types. So we can actually tease out the severity types in this group with different modeling platforms. But anyway, so it looked really useful, and we're applying this to different fatigue conditions. So could we use this to look at mitochondria and also maybe drugs in the lab? So we've done quite a lot of work in the last 6 months to take this forward. So this is the questions, can we see drugs? And can we determine the functional state of mitochondria using Raman? And so this is some compounds. So we've got about 16 compounds we've been looking at as part of this aging project. So there's a couple of examples here. Many compounds have quite nice, fairly unique spectra, which you probably could distinguish from biological materials in a sample. The machine learning has proved really powerful for the PBMC, so I'm quite confident that if we had a combination of these things, we could probably work them out. And in terms of the mitochondria, I don't know if you can see this, there's a little tiny green dot here. So these are mitochondria that we've isolated from cancer cells, and this is a single one, and it has a distinct profile. So cytochrome C has a well-known Raman spectrum, and you can see the peaks here. There's 4 peaks in this sample. So you've got the silent region, the more material, and also this other region for biology. So what we've done is we've used some existing technology in the lab. My lab work a lot with oxygen sensing probes that measure mitochondrial function. So we took mitochondria from a cancer cell, and we basically made them do different things. So here we've got them either poisoned with antimycin A, so they don't respire and don't consume oxygen. We've got them on succinate, just a fuel, so you get the blue line. And if we add ADP, make them make ATP, we get an increase in respiration. And we can also uncouple with an uncoupler to make them work really hard but not make ATP. And I've just taken two of these here. So we ran these assays. We took the mitochondria out at specific time points in the linear phase here and put them onto the Raman grids. And this shows quite nicely. So this is succinate and ADP plus succinate. So the only difference is these ones are making ATP, and the cytochrome peaks are the ones in blue, the blue arrows, to give us some sort of focus. But there are some clear differences you can see in the red. Here, there's a big change here in this peak, and over here, there's a big change. And there's many, many other differences within these profiles. So I'm pretty confident we should be able to see these things, and we may be able to see them in a functional state. And really, just to finish off, really, this is quite a short talk for this meeting. So can we detect drugs inside mitochondria, inside cells? And this is where we are now. And we're just about to start running these experiments in the cancer cell models, which we work quite closely with. So this is an example of one of our compounds, which we use in the cancer space. It's got a targeting molecule that targets mitochondria. So this big peak here at 1000 is linked to this triphosphonium group. So we're pretty confident this gets into mitochondria. So can we see it? And so we'll use the cancer models initially as the models to test the systems, look at the mitochondria. And then we've got some other exciting collaborations with Lynn Cox's group, with the Werner syndrome, Brent Ryan, Parkinson's, and Fran Platt on the 9-MeM-Pixie. And we've just ordered the mice for this aging study to look at, look at monocytes, macrophages from mice at different aging conditions to look at the mitochondria and see how they change. So that's me. So I'll switch off there. We've got a booth at this meeting. So we're happy to ask questions there, or you can contact me if you've got any further questions. Thank you. Oops. I should have stopped sharing. Leo, are you there?
Hello, sorry. Thank you so much for that, Carl. That was a really fascinating story, and I think it's really clear to see how much potential there is, and just I mean, that was quite the tour de force. So please, I would also encourage the audience to take up Carl's offer of continuing the conversation. So we'll move on now to Jessie Coker. Jessie, we're joined by Jessie, who is the Science and Strategy Associate with Action Against Age-Related Macular Degeneration.
So yeah, the talks so far have been quite pre-clinical, quite exciting, and I'm going to make it much more clinical and a little bit more big picture today. So I'm going to introduce you to AAMD's project on pharmacoepidemiology in the open, where in collaboration with UK Spine of Birmingham, we're trying to find new targets for AMD. So AMD is the second leading cause of sight loss globally, behind only cataracts, which in the developed world is treatable with a fairly simple surgery. A full 50% of blindness in the UK is caused by AMD, and 1 in 4 people over the age of 75 suffer from the disease. Driven primarily by an aging population, the number of patients with AMD is expected to grow to 288 million by 2040, making it one of the most costly and burdensome age-related conditions. Zooming in on the UK's 8.7 million AMD patients provides context about the trajectory of the disease. The majority of AMD patients, full 8 million, have what is early or intermediate AMD, meaning that their eye has some preclinical biomarkers of the disease, but they're not currently experiencing any sight loss. Later today, AAMD's CEO Lee more detail about our work on developing sophisticated machine learning models to detect retinal biomarkers to predict the late stage. Once you get to this late stage disease, patients manifest 2 different phenotypes of AMD, one called wet or neovascular, and the other called dry or non-neovascular. The majority of patients suffer from dry AMD, and there is no therapeutic intervention for these patients. The minority of patients suffer from wet AMD, and these patients are often treated with anti-VEGF injections, which have been a game changer for this small subset of patients. However, if you look across the whole disease, from early and intermediate to also late stage dry AMD, the vast majority of patients have no therapeutic interventions. And this stands in fairly sharp contrast to the focus within the research community on late stage disease. 95% of the research initiatives in AMD are focused on firefighting late stage disease, and a lot of these are focused on Me Too anti-VEGF indications, which, as we know, only treat a small subset of AMD patients. So as a charity, we've identified this as a huge gap in the market, and we're attempting to drive focus and research on treatments which might prevent progression of AMD to stop sight loss in its tracks. Obviously, that's easier said than done because AMD is an incredibly complex disease and your eye is a very complex. The macula, which is the portion of your eye which degrades during AMD, of your eye within the retina, requires the collaborative work of 10 different cell layers to detect light and transmit the information to your brain. Much work in AMD in the past has been at the retinal pigment epithelial layer, which is a single cell layer that separates the vasculature from the light sensing portion of your eye. There's also been some work recently in AMD, choroid, which is the name for this dense neovasculature network that feeds the eye. However, if you look from a systems biology approach, depending on this level of your eye you look at, different pathways seem to be misregulated. Choroid and the retinal pigment epithelia cells, you see changes like overactivation of complement, degradation of the extracellular matrix, misregulation of autophagy, which fits nicely about what we discussed earlier, as well as changes in glycolytic flux. As you move up into the light sensing portions of the eye, things like hyperactive RTK signaling, a change in the transport of nutrients, and misregulation of lipid metabolism also appears. When you look at this in a network analysis, you can see that there are hundreds of different proteins and genes implicated in the progression of AMD, and that the driver genes change depending on the cell layer you look at. So this makes it both incredibly difficult to pick a target and also incredibly difficult to pick a phenotypic assay model. So as a charity, we're also limited by financial resources, and we can't plow hundreds of millions of dollars into a single preclinical drug development platform. So as our goal is to get a treatment to AMD patients in the near-term future, we've started our work by focusing on drug repurposing. So to start, what is drug repurposing and why is it helpful? Well, because the drugs you're looking at have already been approved for use, the safety of the molecules is demonstrating, allowing you to simplify your clinical trials and usually begin work at phase 2 efficacy studies. Additionally, drug repurposing workflows can be faster because a significant fraction of the preclinical work is bypassed. And the work can be both cheaper for the research group and cheaper to final patients because the drug can be used off patent or as an additional use. Now, sort of typical in vitro drug repurposing relies on taking a library of hundreds to thousands of FDA-approved drugs and analyzing them in some type of phenotypic assay platform. As these assays become more representative of human biology, for example, in 3D tissue culture or organoids, they become more expensive to run. And especially for a disease like AMD, which requires this kind of symphony of work between different cell layers, even the most representative phenotypic assays are quite reductionist. Finally, with in vitro drug repurposing, the utility of your molecules in patients is an open question. For AMD, we've been trying to look in a different type of repurposing, which we've nicknamed in hindsight, which relies on pharmacoepidemiological studies backed by big data. In this approach, you take a database of patients who have been treated with a variety of different drugs, and you look to see if any of those drugs reduce the risk of progression to AMD. With in hindsight drug repurposing, there's no model system, and the utility in human patients is the direct research question. Finally, because most of this upfront work is done in silico, in hindsight drug repurposing is low cost and can be done quite quickly. For example, the project I'm presenting today from start to finish will only last 8 months. So the mission of our project then is to leverage in hindsight repurposing to investigate whether any approved medications are associated with a reduced or increased risk of AMD. Our goal is to identify novel targets, priority mechanisms, and potentially repurposable drugs. And as a charity committed to open science, we will immediately share the outcomes of this project with the research community, both on our website and within academic publications. We've set up this project as a true knowledge exchange. AAMD, as the governing charity partner, brings disease insight, a commitment to open science, and an explicitly translational objective. All of the pharmacoepidemiology expertise, as well as database access and computational resources, is coming from the University of Birmingham. And AAMD and the team at Birmingham are working together monthly to set project objectives and to define analysis goals. Finally, at the end of the stage of the project, we'll be hosting a community hackathon where disease experts and ophthalmologists can come together to provide insight about target biology and to help transform our hit list of potentially repurposable drugs into true therapeutic hypotheses. This community hackathon will be taking place in July 2021. And if you're interested in being part of this event, please reach out to me at the end of the talk. So in conclusion, we're going to apply machine learning and statistical models to a database encompassing all drugs that have been prescribed in the UK over the last 20 years to look for any which reduce the risk of progression to AMD. Following expert review at a community hackathon, we'll be releasing to the community a hit list of drugs for clinical trials, potential protein targets for optimization, including for retinal delivery of the modifying molecule, and finally, potential and priority pathways for modulation in AMD. Furthermore, we hope this project sets an example for a new vision of charities beyond simply giving cash to a project. At AAMD, we're committed to active co-development of science and the setting of objectives at the charity level. And we hope that this project can set an example of how a true knowledge exchange can be formed between research scientists and a charity partner. With that being said, I thank you for your time. Both myself and AAMD can be found on Twitter if you'd like to reach out. Thanks very much.
Thanks, Jessie. That was really exciting work. So we'll move on to our last speaker of the session. And we've got Phil Atherton, who is the Chair of Clinical, Metabolic and Molecular Physiology at the University of Nottingham. And Phil's going to tell us a little bit about a project focusing on the integration of omics into musculoskeletal decline in aging and disease. So if you're happy, Phil, I'll pass over to you for the last talk.
Hi everyone, just waiting for the opportunity to share my screen. Sorry, there's another settings issue, just bear with me. Leah, at the moment, I need to probably close this. I've tried to sort all these settings issues, and there's another one now for me to share screen as well. So I'm going to have to probably exit for temporarily. My apologies.
No problem, Phil.
I'll just go into my security settings and see if I can alter it. I can't do it as the opportunity came up. It wouldn't allow me to. I'll see if I can quickly correct it. It might need a restart again. I've done this 3 times already with different settings today.
Sorry, Phil.
It's all right, but
If you need to restart, Phil, then
yeah, I might have to. Sorry, it's
okay.
just I need to restart. It's not letting me check the box for the next screen share now.
Okay.
So bear with me.
No problem.
If you need to reschedule, Leah, sorry, and move on and slot me in later while I resolve it, that's fine.
We'll take a question. So
okay, give me a second. Sorry, best I can.
Right. So Carl, could I direct a question to you if you're there?
Yep, sure.
So we've had a question about the use of the Raman spectroscopy. So will the interrogation of mitochondrial function potentially alter the progressional path and the development of interventions for aging? Or do you think there'll be any impact on the assessment of existing candidates?
That's quite a bit of questions there.
Yeah, sorry.
I suppose for screening point of view, so you could screen things in vitro, maybe with the technology, and also running things in animals. It would be a lot easier to look at samples with this approach, bits of brain tissue, or to get a real handle of what the mitochondria are doing. You know, somebody talked a bit earlier about the early stages of some of these problems like Alzheimer's and dementia. You do see a problem with glucose metabolism very early in some of these conditions. So it may give you a handle on some early changes in aging models, and if you can actually interact with those, then yes. In terms of affecting the aging space, I suppose it's quite hard to get biopsies from people. So maybe the PBMCs could be a useful window. We were very surprised to see such big differences between multiple sclerosis patients and ME. You know, there's no test for ME, and we can really distinguish from the controls. And why the multiple sclerosis patients also different, but in a different way. So there could be markers in the blood that you could look at. We're just about to start looking at plasma and cerebrospinal fluid from patients with MS if we get the money. And so maybe that might be a way into the brain to look at what's going on in the brain by taking a small bit of fluid. So there's definitely potential there. And the PBMCs are tricky. You've got to make them. They're quite easy, but plasma or fluid would be much quicker. There's different machines we can use. It takes 30 seconds to analyze a sample. So it could be very, very, very interesting.
Yeah, that's really promising. Jessie, I wonder if I might ask you a question. I'm going to be as provocative as I was earlier. Could you give us any sort of educated guess as to when we might expect to see some of these repurposing drugs coming through? What's your feeling on how close we are to having something we might be able to try in patients?
It's a good question. At earlier stages of a similar project, we've taken some of the existing hypotheses from the literature about potentially repurposable drugs in AMD, and we've kind of applied a more focused pharmacoepidemiology study to them. So just doing one drug at a time. And some of those results are under review now. Without going into specifics, I can tell you that some of the things that have been floating around as good ideas don't seem to bear out when you look at patients specifically. So this is one of the reasons why we're doing this sort of unbiased approach, where we're looking at all the drugs in the existing UK databases to see if there's any signal. So it's hard to speculate as to when they'll start reaching a clinical benefit, but I can say that the results of the project should be out for the community well before the end of 2021. So at least that step will be taken, relatively soon.
And in general, as a quick follow-up, do you think there's, do you sense that there's lots of excitement in pharma and industry to look at repurposing?
Pharma and industry, I would say probably not. Almost by definition, it's something that charities prefer because repurposed drugs are not the big money makers that pharma focuses on. And you can see in AMD, most pharmaceutical work goes on anti-VEGF injections, which are already proven to be beneficial for a subset of patients. So especially for this kind of early to intermediate AMD and a prevention of disease, which is our mindset as a charity, there's very little work going into it from the industry, which is a gap that we see and are trying to address.
Great. Okay, Phil, you're back with us. Do you want to give it another go?
We can try. I mean, I don't know if you can hear me, but I've tried everything, and there is seemingly no way I can share this screen.
Phil, do you want to send it to me? And do you want me to share them for you?
Anything would do.
Try it. Send me an email, and I'll try and share them for you.
Sorry,
tell me when to
everything. I've never had this before. All the settings seem fine, but I can't share the screen. There's just a cross through, and everything. All of my settings suggest that there should be no admittance problems. Right, Carl, I shall do that now.
Okay.
Felix, may I ask you a quick question? So you might be able to see this in the chat. We've been asked, how might the concept of free fatty acid supplementation to support shifting towards anti-inflammatory macrophage phenotype be translated into clinical intervention?
It's a very good question. I think it's probably sort of like about mostly trying to shift sort of like serum levels towards fatty acid species that are sort of like known to, for instance, reduce risks of cardiovascular diseases. So some of this is really probably the most simplest idea of it is really dietary supplementation. So specific types of foods are just enriched in these free fatty acids, such as like vegetable oils typically, or soya beans. Are typically quite rich sources, for instance, of these kind of like polyunsaturated fatty acids, to which, for instance, linoleic acid belongs to as well. And there's, I think, now slowly also an increasing body of evidence as well, suggesting sort of like that these ones are kind of like quite good for you typically, and kind of like can calm inflammation.
Okay, so Tech have told me that if I leave, you should be allowed to share, Phil. So I'll hand over to you.
Maybe. No. No, the share option just isn't working. Oh, hold fire. Yeah, thank you. I think it's now crashed my computer is the problem. I can't even open PowerPoint. Here we go. Hold on. Can you see this?
No, not yet. I can't see your email either. I don't know where it's.
Yeah, it's up on my screen now. So I've it's just, I think it's basically crashed my computer. So all I can suggest is, Carl, if you can just
I can't. It's not come through yet, Phil. It's not come through for me yet. I don't know.
Try once more. It's disappeared. The options come back again. Let me just try once more. See if I can get it to work. In the Chrome tab.
No, it's still not come through this end. Maybe it's not.
Can you see now? Let's see if this works again.
It's showing us something.
Still no, you still can't see the presentation.
Well, I can see your desktop maybe, or somebody's desktop.
The presentation's up for me. It's just you can't see it.
Oh, right, okay.
Yeah.
Is it a big file, Phil?
No, it's not, Carl. It shouldn't be. It's sent to you. It should be really small. It's 9 meg. That's all.
Oh, right. That should come through. Oh, got it.
Great.
Okay, I'll try.
Thanks, Carl.
Okay. All right. I'm trying to share mine. Can somebody let me do it? I've got a grayed out button. Yeah, the button's still grayed out on the share option.
Okay, we've actually got a space tomorrow, Phil, so we'll reschedule.
I think it makes sense. I've no idea what's going on, but I've never had this before, but there's no way I can do it via Chrome on this Mac at the moment.
Can you see it now?
Yes, but not full screen.
I can see it. It's up to you, Leah, if you prefer to.
If you're ready.
Okay, yeah, sure.
Okay.
So
thanks everyone for your patience.
I'll keep this really briefly here anyway, and for everybody, and just give the synopsis. So thanks, and I apologize everybody that, it's been a nightmarish year anyway, and so we don't need this, but my project I'm going to talk about briefly today, or extremely briefly now, is entitled The Impacts of Mechanistic Target of Rapamycin Inhibition on Aged Muscle. And while this is a muscle related talk and it's very brief, one of the things I'll highlight is that is in keeping with a lot of the spine stuff and comorbidity. It's really a pathway that sort of speaks to many cells beyond muscle. And so the derivation of these sort of ideas came far before those specific to muscle. Carl, if you wouldn't mind, if you could switch to the next slide. So here's an overview of the background of mTOR in relation to cellular aging. And this is an illustration that I took from a review article published a couple of years ago. And there are many out there, by the way, that sort of illustrate very similar things. Putting it briefly, mTOR is a very ubiquitous pathway in cell types, and that's why it has such potent effects across cells. So whether it's the immune system, the musculoskeletal system, or cardiovascular system, and also as it pertains to aberrance in disease and disorders, it becomes quite an important pathway. And it's one of those that you probably couldn't find an area that you wouldn't be able to find some important degree of regulation or dysregulation in health and disease. So if you look at the left-hand side here, you can see some of the main processes that it sort of regulates. So you've got mTOR in the blob of the yellow in the middle. It's a key regulator of protein synthesis within cells. So it responds to growth factors and nutrients, in particular carbohydrate and amino acid availability, and in which it can regulate proteostasis, in other words, muscle protein synthesis, muscle protein breakdown, the same in other cell types too. By the same token, actually, by activating TOR pathway, you actually induce inhibition of autophagy. And now in youth, that's seen as being a positive thing. So in other words, you sort of have the vitality where you promote the growth of cells and you promote the inhibition of the degradation of cells. But if you look at the box in the middle, the problem is that if this activation of this pathway becomes excessive, it can promote things that are damaging and which are quite ubiquitous across aging cell phenotypes. So DNA damage, cell stress, DNA damage accumulation in both DNA, proteins, and even lipid species, and also, as Carl's mentioned before, about mitochondrial dysfunction potentially. So the links for this pathway's activation has been shown in many different conditions: cancers, malignancies, cardiovascular disorders, neurodegenerative disorders and diseases, but also things like stem cell function and tissue repair, which of course are interrelated. So the main theme here really in a nutshell is that the sort of hyperactivation or overactivation of mTOR, which is stimulated through lots of different means in different cell types across the body, is somehow linked to tissue degeneration and the propagation of different disease states across cell types. And the question we had is in relation to muscle and sarcopenia, which is the or frailty, which is the age-related muscle wasting and dysfunction. Carl, if you wouldn't mind to switch to the next slide. So this is just the background in terms, it's quite heavy in text, so I'll summarize it quickly. Age-related sarcopenia, a loss of muscle mass and function, is associated with frailties and falls. So it's one of the sort of main indicators of poor health span. There are no strategies that exist that are effective, drug that is, other than exercise. And exercise actually has more limited effects in aging than it does in younger populations. Here we're actually proposing in the Spine Project the repurposing, which the previous speaker talked about on a larger scale of discovery of a long-established compound, which has been shown in animal models, both to extend lifespan and also to limit muscle decline. And protect aspects of health span as well. So it's kind of one of those ubiquitously acting compounds that seems to affect multiple cell types, which probably underlies its impacts on longevity and aspects of health span in preclinical settings. The compound in question is called Rapamycin. It targets the mTOR pathway, not necessarily entirely specifically, but it is naturally occurring compound and it's phase 2 safe. So it's not been trials haven't been conducted for many different conditions, or they have been for some, looking at various different endpoints, again, given the fact that TOR is such a ubiquitously acting pathway. But nonetheless, one of the things that caught our eye was it over a few months period, its use was phase 2 safe. So that kind of gave us the impression that we could repurpose this drug potentially for the use in muscle and sarcopenia. Going back to muscle then, so while mTOR usually helps maintain healthy muscle, I mentioned in the first slide that positive signals through this pathway would normally be associated with propagation of growth and limitation of degradation, let's say, of muscle tissue protein. In aging, it seems it becomes overactive. That might be deemed a good thing. The problem is that when it becomes overactive, it can sort of bring with it other aberrant functions, such as, again, the limitation of autophagy by function. In other words, that means that if you limit autophagy, you can get build up of things like ER, so endoplasmic reticulum stress, malfunctioning proteins and organelles and the like. So our idea was to take this into people and to try and understand a little bit more about how inhibition of mTOR in vivo might actually affect skeletal muscle function. And Carl, please, if you just go to the final slide. Thank you. So this is our protocol. Again, we've got a booth there, so I'd be happy to answer any questions on this. We have a 2-arm approach. We have a placebo versus a Rapamycin, which is a rapamycin, approved rapamycin drug. You can see the oral dosing there. We're going to conduct this trial on a small number of individuals, older individuals, for 4 months, which was the equivalent to the phase 2 trial, showing relatively few or minimal or no serious, at least no serious adverse effects. We have a battery of outcome measures, which are shown in the table below, and they include aspects of muscle mass and function, blood sampling, where we can look at lots of different parameters, including things like immune function, the usual blood biochemistry associated with health parameters of different organ systems. Then we're obviously looking directly at muscle biopsies through sampling muscle from the leg, in which, using techniques we've developed, we can look at the turnover of substrate pools, which would tell us about things like mitochondrial, myofibrillar, and overall muscle proteostasis, functionality in response to the drug. And also we'd be looking at neuromuscular function, because we know that some of the apparent protective characteristics of rapamycin-like drugs on the neuromuscular system relate not just to muscle, but the nerve. And the final slide is just acknowledgements, and I'll be happy to leave it there, and apologies for all the technical issues. Thanks, Carl.
No problem.
Thanks ever so much, Phil. Thanks to the audience for sticking with us. But I think that it's been really, I'm really excited to see what comes out of the trial, and how the different battery of measures, what you find on those. So we'll wrap up the session now. Please do, as Phil's already said, go into the expo booth to direct any questions. And of course, as I've said, all the speakers are contactable directly through Hopin. You can send direct messages. So thanks so much to everyone for coming to this short talk session. We'll be back in 20 minutes, about 15:15, to have our last session of the day, which is our panel session, which will be moderated and chaired by Professor Janet Lord. So please join us for that. It's going to be a really nice session. And I think we must have all of the technical bugs out. So we'll see you again slightly later on. Thank you.